All right. Good evening, everyone. I hope all of you can hear me and also see the screen. We are restarting this webinars after a gap of almost 1 month. Yeah, this live session on YouTube is happening after almost a month. All right. So thank you so much for joining in from different parts of India. And I'm also sure you're watching, some of you are watching on YouTube as well. The recording will be available on YouTube, so don't worry about that. Please pay attention to this webinar. The objective of this webinar is to create awareness about formulation and development as an important career path for pharmacy professionals. And why is this very important? Because this is probably the only division in pharmaceuticals where only pharmacy professionals are preferred, right? So it's an important one, and we should be aware of what is happening here, right? And with us is Santosh. Santosh happens to be from my college, the college where I studied. We studied together. Santosh, if you can switch on the video, let's see if you are there. All right, perfect. Good to see you. All right. Hi, Santosh. So Santosh, hello, Santosh. So Santosh and we studied in the same college. All right. And Santosh has been working in this world of formulation and development for the past 13 plus years, 13 years plus, right? Santosh did his B.Pharm from Government College of Pharmacy, Bangalore, the same college where I also did B.Pharm. And then he did his M.Pharm from Sri Siddhganga College of Pharmacy, Tumkur. Right. He comes with a rich experience in formulation and development, 8 years in allopathic formulations, and then about 5.5 years in Ayurvedic formulations. Currently, he's working as a formulation scientist at Himalaya Wellness Company. And Santosh is a proud pharmacist. He believes in helping other pharmacy professionals and help them choose F&D as a career path. And that's where we got connected. So I requested Santosh to create that awareness to our pharmacy professionals about formulation and development, and also help us understand how formulation and development can be a niche area for many of the pharmacy professionals or students, and how one can choose the path to formulation and development. So Santosh, welcome. The stage is all yours, and you may continue from here, and let's see how much we can learn from today.
Thank you, sir. Thank you very much for giving the opportunity as a speaker on formulation development.
My pleasure.
Yeah, now the formulation development. Formulation development is a core of pharmaceutical industry. As we all know, what is formulation development? The name itself will indicate developing a right, means a robust optimized formula along with robust optimized process to get the quality of product. So what is quality of product? Yeah, before going to this, I will just introduce myself. Myself Santosh Jia. Yeah, I studied my B Pharmacy in the Government College of Pharmacy as self-taught, and I finished my M.Pharma in Siddhganga College of Pharmacy, Tumkur. And even I am also from village background. I was also a little poor in English at the time. So, like few people, I am also intentionally, even me also not join pharmacy. Actually, I supposed to join engineering something. So that time there was a CET results were postponing, counseling. So it has postponed some 2-3 times. So in my home, my parents are told, "Oh, it's postponing all the other college. The admissions and are closing now. You better, for safer side, you first join on government science college. Anyway, the fee is less. So we'll see if you get any good college so that you can join engineering." So I joined government science college. Then I got some information that there is a pharmacy course is there. It's very good. you can join that level. I got some information from my friends. Then I did some research there. They were told that you will get very good job in even in outside of India. It's having very good scope. So usually I used to do something different. So what I did, I just applied for pharmacy course. Before that, I also applied for BSc Agriculture and Veterinary also. So during the counseling, I just went. I was not knowing any colleges about pharmacy. Which college is there? Is there any government college? How many government colleges are there? Even I don't know anything. I just attended counseling and I asked them, sir, I'm staying in Nelamangala. I want some college, any one college that is near to my place. So he showed me government college. Even I was not knowing government college also. I'm searching the address itself there. Some Kalingrao Road is mentioned. Sir, where is Kalingrao Road comes? This is the government college, only one college in Karnataka. You tell first if you want, I will click it. Otherwise, you won't get. Then finally, okay, sir. So I took that college. The last date for admission is, suppose you consider it is 7th. My CET counseling will be on 8th. If it is vice versa, then I can do something at least. So what I did, I visited government college once before admission. I visited and I saw all the facility and all. I met one senior person, one senior student. I asked him like this, how the college, how the teaching. He told what you are doing. I told I'm waiting for CET counseling. I don't know about pharmacy. I want to join and I want to know OS property. Because you don't need anything. You first join it. So then I went home and decided to join. When I joined, I didn't attend it. We see to cancel in the next lesson. So after that, I finished B Pharma. During B Pharma, because why I'm telling means the same thoughts will be going on in people's mind also. While I'm doing B Pharma, I thought I will finish in Pharma and I will do PhD. And I will join for lecturing because I was little interested towards lecturing in the teaching center. So I thought like then I joined in Pharma. Still in Pharma also I wanted to do PhD. Then my lecturers itself told, what you will do after PhD? I told I will do lecturing. In lecturing and all, usually you won't get very good salary in compared to industry. You just think it up. To finish PhD and all, it takes 5 to 6 years. So I'm not telling PhD is not good. I'm just telling how my field has changed to industry. So then I thought, okay, because again after the informal, if I wait for 5 to 6 years means it's very difficult. I have to have it. So after Informa, then I was thinking, during Informa itself, I was thinking in industry, which department I have to go. So everyone knows, even at the time also formulation development, because I am doing Informa in pharmaceutics. But formulation development, people are telling, hey, it is saturating there. It's very difficult to get in R&D. Then I thought, let's try. Let's try first. Let's prepare myself for the formulation development. Suppose if I won't get formulation development, what is the next option? During BPharm days, I felt the difficult subject was for me the jurisprudence, means rules, regulations, and all, because we are full in English. So we find difficulties to understand this jurisprudence. Right? So I thought this regulatory means what? Regulatory means it is a regulate. You have to know the regulatory rules and regulations of particular market, the regulatory bodies. You have to know. You have to work in that. Then I thought I was not knowing what is regulatory bodies. I just know that regulatory body means each country or each market has its own regulatory regulations, laws, regulations for particular products. So I thought, it won't suit me. Let's keep for last. So what to do after formulation? Second option, quality assurance. Okay. In production, related. Like that I thought. So luckily I got some opportunity in formulation development when I joined Chemwell Biopharma as a formulation development in solid orals. Then after to some tutorial experience, I joined myself as worked in Harris West. Then I came back to herbals, Ayurvedic formulations in a way. So this is my story. So now let's talk about formulation development. What is formulation development? As I already told, formulation development, the name itself will tell that development of robust optimized formula and robust optimized process to get a quality of the product. So what is that quality of the product? It includes all these things. Better shelf life. Better shelf life means what? It has to withstand.
Santosh, sorry, your audio is not very clear, it seems, to some of the participants. Could you just see if your microphone has something to do with that? Try speaking a bit closer.
Okay. Is it now audible, sir?
Please confirm, guys. Is it clear now? Not mine, of course, Santosh. Audio. Santosh, you try speaking something.
Is it audible? All of you?
I think it is better now. Yes, please go on. Thank you.
Okay. So what is quality of the product? Quality of the product means the product should give better shelf life. Means it should withstand at different temperature and humidity conditions, and during usage of the medicines, during transportation of the medicines, and it should not have microbial contamination or microbial growth, and it should give very good therapeutic efficacy for a particular formulation. Means it should give very good bioavailability. If it is generic, it should give very good bioequivalence. Then it should have optimized formula and process. What is that optimized formula and process? Optimized formula and process means a slight changes in formula or process should not affect the quality of the product. Slight changes means what? See, in formula means we are using so many raw materials, means excipients, actives. There is little variation, batch to batch variation, or manual errors. And the process means what? There are changes, slight equipment changes, models. Again, the manual error. So there are some, we have to formulate in such a way that we have set a range. Within that range, if you formulate, that should not affect the quality of the product. So we have to optimize that type of formula and process. Then our formulation should be patentable. Suppose if we are doing formulation, or our formula or our process or our claims should be different from marketed products, then we can apply for patents also. Again, it has so many criteria. And competing enough in market. Competing enough in market means our product should have novelty, and our product should be customer reachable. It should be easily reachable to customer in terms of price and in terms of novelty. So this all includes formulation development. So what is the scope of formulation development? Scope of formulation development means the product, whatever the product in the market, should be developed by formulation development person. So that's why I said it's a core of pharmaceutical industry. In formulation development, only M.Pharma pharmaceutics can enter. M.Pharma students who has pursued pharmaceutics department, they can only join formulation development. No other candidates can compete there. But again, there are little drawbacks also is there in the market. What drawbacks? See, in India, most of the pharma companies, they are not doing NDA. NDA means in terms of new drug products, new innovations. Most of the pharma companies, they are doing generics. So almost generics, how many companies will do generics? 10, 20, 30 for the same product. So these type of saturations are occurring. But at the same time, I'm not telling it is not down. So for formulation development is the main core of pharma industry. Then let's talk in detail at the end of the session. We'll discuss first on formulation development. What is formulation development? Types of formulation development. What are the stages involved in formulation development? Then we'll discuss on career paths. So types of formulation. As you all studied, there are solid dosage forms like tablets, capsules, oral powders, liquid dosage forms, syrups, suspensions, etc. Semi-solid dosage forms, ointments, parenteral preparations, injections, serums, etc. So, steps involved in formulation development. What are the steps involved in formulation development? First, if you get any product to develop, if you want to develop any product, you want to do some literature search. What is that? What solid dosage form? What is the solid dosage form of the product? Which market the product is going? What are the regulatory body? Is there any patents available? What are the regulations for particular market? What type of excipients I have to take? I have to choose. What is the dosage of that excipients? All these literature search you have to do. Then based on literature search, you have to select the excipients. You have to select the excipients and its dose. You have to select the process involved in particular dosage forms. You have to make down all the points for a particular regulatory body. Then you have to do pre-formulation studies. What is pre-formulation studies? Before formulation, you have to do some studies on active ingredients. For example, you have to study the active ingredients, hygroscopicity, whether the active ingredients is hygroscopic or non-hygroscopic, whether it is soluble or insoluble, whether it is permeable or highly permeable, low permeable, as per BCS classification is there. So what is the particle size of the active ingredient? All these things. By getting all the information of active ingredient, you have to do that compatibility study. So suppose if you are doing any generic product, you have to study the standard, means the innovator product also. What is the innovator for this? What claims they have did? What are the excipients they have used? What is present in patient information leaflet? All these things. So now come for comparative study. Comparative study means what? You are going to ensure the compatibility between drug and excipients, drug and drug. Drug means active ingredient, active pharmaceutical ingredient. API means active pharmaceutical ingredient. Drug and drug interactions. Drug and drug compatibility is there or not. Drug and excipients compatibility is there or not. Excipients and excipients compatibility is there or not. So how to study comparative study? I won't go in much detail. I'm just briefing you, just giving an idea. What is formulation development? So in compatibility, what they will do? They will mix the active and excipients at a specified proportions, and they will charge it under different stability conditions or environmental conditions like 40-75, 25-60, 30-65. These are all the environmental conditions which are available in different corners of the world. So compatibility study they will do for 1 month. What and all study they will do, what and all the parameters they will check there. How they will ensure that there is a compatibility between actives and excipients, actives and actives, excipients and excipients. So there are physical and chemical parameters are there. Physical what parameters? They are going to check, is there any change in color? Is there any change in appearance? Is there any what is that? Is there any there is a transparency to the color change from transparency? Suppose if it's liquid from, is there any turbid is there? And what are the chemical changes they will do? They will check the assay. If the assay is going to be less, the product is going to be degrading. Or is there any generation of impurities? All those things they will do. And one more thing, is there any polymorphic transformation of actives is happening or not going to by scanning electron microscopy? So, and also there are two methods are there. The methods are non-thermal techniques and thermal techniques. What is non-thermal techniques? IR, near to IR, and Raman spectroscopy. These studies they will do. What is those things? IR, near to IR, Raman spectroscopy, and all. They will provide unique fingerprint on particular excipients and on particular actives. So if there is any changes in the mixture of samples during compatibility, they will find out. And also they will do differential thermal analysis, that is thermal technique, by using differential scanning calorimetry. There, if any changes in the sample can be found out by exothermic and endothermic, by comparing the inert excipients in single excipients or single active. So, is there any transformation of excipients in the mixture of samples like glass transition, sublimation, melting point, crystallization? All these data can be provided by differential scanning calorimetry. So by these data, we can easily find out, is there any compatibility, is there any incompatibility is there between the active ingredient and excipient, or excipient between excipients and excipients, we will find out. So by this study, we will select the proper excipients. Then we will go for prototype formulation. Prototype formulation means by collecting all these by collecting all these data and information, we will make one tentative formula and tentative process, and we will take a trial. That sample we will submit for analysis. What they will do, there are one more department is there, analytical R&D. Like formulation development, there is one more department is there, analytic R&D. R&D means there are so many departments are there. Right now, I'm going to explain only formulation development, which are linked. There are few departments which are linked to formulation development, that is analytical development. Analytical development, they will collect the samples from us, formulation development, and they will develop the method of analysis. Method of analysis of different test parameters like dissolution method will develop, assay method develop. Examples like impurities method develop, disintegration time, etc. So they will do their job. Here, what we have to do after doing product formulation, we will charge it for stress study. What is stress study? The name itself indicates stress. Means we are putting a sample. We will put a sample at different stress conditions. Means at a very high temperature, a very high humidity, and acidic environment, a basic environment, etc. That we will study for. Usually, we will study for 1 month. Then you will analyze: is there any changes occurred in stress study, physically or chemically? That gives the robustness of the formula. Means if any changes occurred, that gives a confidence to go for stability studies. Because stability studies and all we have to do for longer days. So for each trial, we cannot do stability studies. We have to wait for minimum 6 months. So what we will do? First, we will do stress study for 1 month. Based on that confidence, we will go for stability studies. So before going to stability study, once we get confirmation from stress study, we will, is there any changes? Is there any little changes in based on analytical data? We will again play with some formula and process, and we will optimize the formula and process. And with the right formula, we will charge it for stability studies. And we will wait for stability studies for 3 months, 6 months. If the stability is okay, then we will submit the sample for small bioequivalence study. Why small bioequivalence study we will do? Why? Because instead of doing bioequivalence study after pivotal batch, if any failure happens, it's a huge loss to the company. So at lab stability batches, they will do bioequivalence study. At very small subjects they will do. So once the bioequivalence study also get passed, then we will go for pivotal or scale-up batch. Pivotal or scale-up batch means till now what we have done, we have developed the product in formulation development, that is R&D lab, in small scale. So now we want to see the feasibility and reproducibility of that formula and process in production plant, means which is having higher capacity of equipments and higher batch size. We will face so many scale-up challenges. Means small scale, doing small scale is different, and doing the batches in higher scale is very different. So we will face so many challenges there. Those challenges we have to face, and we have to troubleshoot. We have to optimize it, and we have to make the process more feasible. If it will take on reproducibility batch. That batch you will charge it for stability again. So here I'm telling stability means why they will do stability. Stability means they are doing in different environmental conditions. Different environmental conditions. What is the different environmental conditions? See, the environmental condition is not same in Bangalore, not same in Chennai, not same in Hyderabad. So the environmental condition is varying. Suppose in Bangalore it is very cool, in Chennai it is very humidity, in Hyderabad it is very hot. So that's why our product should withstand in all the environmental conditions. So we will charge it for 4075. That is called accelerated stability condition. Why we call accelerated stability condition? At very high temperature, 40 degrees high temperature, 75 degree humidity, 75% humidity, high humidity and high temperature. That is 4075. We will call it as saturated stability. Here we will do 6 months study. The 6 months study and 25/60, that is long term, 30/65 intermediate. Again, it depends on market. Again, the little variation, the little changes will happen in stability conditions. So once studying the stability study, once we will confirm the stability study is okay, then we will do bioequivalence study. Again, bioequivalence study previously we have did in very small batch. Now we will do pivotal batch bioequivalence study. That with higher subjects we will do. What is bioequivalence study? Bioequivalence study means here we are going to find out the bioavailability of test product with the innovator product. It has its own limits. If the bioequivalence is passed means it has its own limit. Means 90% confidence interval of test product should be fall within 90 to 125 of reference product. So once this bioequivalence study passes, then they will go for exhibit and validation batch. Exhibit and validation batch means, so we have finalized the formula and process in production plant. So that we are going to take 3 big batches and we are to validate the process. What we have finalized it, formula and process, that formula and process we have to validate with 3 batches. The 3 batches should show reproducibility. For that purpose, they will do validation. Whether all 3 batches are validating or not. Whether the formula and process are robust or reproducibility is coming or not. That they will do. That they will do in even formulation point of view and also analytical point of view. Analytical point of view, what they will do? They will analyze the samples of 3 validation batches. They will check the reproducibility is coming or not. Reproducibility results are coming or not. Whether their method of analysis is correct or not. They will validate their methods. Formulation methods, we have to validate our methods. For formulation purposes, we have to validate. And that will charge it for stability. So most of the market, this exhibit batch or validation batch means they will take 3 batches. That is 1/10. Each batch size is 1/10 of the commercial batch size or 1 lakh units, whichever is small, you can take it. And out of 3, 1 batch you can take 25,000 products. But generally they will take all 3 same 1 lakh units only. So after exhibit and validation batches, they will charge it for stability. Again, because again it's validation only. Again, we have to check, we have to confirm that stability study is that our product is more stable in the product. So once the completion of 6-month accelerated study, why 6 months accelerated study I'm telling? Once completion of 6-month accelerated study means they will give confident enough our product is stable. So what we will do? The product and the company also. So common technical document contains 5 modules. I will explain you here. 5 modules. That is module 1 is it contains administrative information. Module 2 contains overview and summary of module 3 and 5. Because the common technical document we cannot write simply one from what and all I will not like that. It has separate subclasses, and each classes will be done by a particular department. So module 2 contain Overview and summary of module 3 and 5. What is module 3? That is quality, regarding quality. Means pharmaceutical documentation, how you have developed the product, the complete information for the stability, how you did stability study, how you did stress study, how you did dissolution, how many multimedias you have used for dissolution, what is the discriminating media, how it is coming, what is the assay, how it is coming, impurities are within limit or not, how I develop it for the product, what is the clinical data, what is the bioequivalence data, everything you mention. But the clinical data, there is a module 5 is there. Module 5 contains complete clinical study reports. Module 4 contains non-clinical reports, means toxicology, pharmacological studies. Clinical study means safety study they will do. And bioequivalence study means that they will do mainly for generic drugs. So all this information you have to make it in a proper way, and we will give it for regulatory department for reviewing them. They will go for filing. Once the filing happens for a particular market, the product will be launched into the market for particular market. This is formulation development. I just explained you in brief. So here technology transfer activities. I already have explained about technology transfer activities also. So I can tell you one more thing. Technology transfer also one department. Previously, what they are doing, formulation person itself is doing technology transfer activities. What is technology transfer activities? Technology transfer activities. We are transferring the technology of R&D to the production field. Transferring the technology of R&D to production field. Means they will be coming in between formulation and production. So till R&D, what and all we have developed the product in R&D level, that formulation person will do. Once formulation person will develop the product, the technology person will come. We have to show our product to the technology person, how we have developed, what is the process involved, and they have their own limits. They will ask whatever the queries they have, all the queries they will solve it. They will ask all the stability study. They have the optimization study. So after everything, formulation person and technology transfer person both will do technology transfer activities. Means that is scale up and pivotal batch iteration, the scale up and pivotal batch, and exhibit and validation batch in production plant done through technology transfer person. These are technology transfer activities. Again, if you go in depth, there are so many is there. If you go to the guidelines and all, I'm just giving a brief idea. Career path in FD. Career path in FD, formulation development. As I told, only M-Pharm persons can only, M-Pharma in pharmaceutical background persons only can enter formulation development. Even B-Pharma persons, nowadays, no industry is taking any graduate persons. They are asking all post-graduation persons. M-Pharma in pharmaceutics. So there is specialization. So there is no any other person can compete for formulation development. Only purely M-Pharma pharmaceutics person can only do it. Can enter formulation development. So how much the scope to come to the salary and all, generally I will speak in and out generally. That's what we are seeing in India. As my experience and as what we have heard, it's around 2.4 to 5 lakh for the freshers, depends on company. So Indian average you will get 3 in India. Eligibility, as I told, only pharma and pharmaceuticals person can go into the formulation development. Skills. What skills are needed for formulation development? as we discussed, all these skills and all needed. But I know all these skills you won't come to know without entering the industry. But at least you people should know different dosage forms. You should have a little bit knowledge on raw materials, excipients, and its functions. Minimum basic knowledge on formulations, basic knowledge on regulatory bodies, little bit information on bioequivalence study. You can easily, I will tell you one more thing. You can easily, if you want to read all these things, you can easily search in ICH guidelines. You can easily get in all these informations. You can just search in Google ICH guidelines. You will get a link and you can open ICH.org. The website is there. All the guidelines we have to do. You know, if you just read it also, you won't understand it completely, because without experience, it's very difficult to imagine and understand itself. Because so many words are there very new to people, but still there are nowadays you can get in Google, YouTube, so many ready informations. You can learn it. So it's good. You should have all this knowledge, technology transfer activities, bioequivalence studies, regulatory study, regulatory information. What is the regulatory body of US FDA? What is the regulatory body of Australia, TGA? What is the regulatory body of South Africa, MCA? Like that. If you have all this information, at least minimum informations. What is the difference between the European and US FDA? What's the difference between European regulatory body and US FDA regulatory body? What is Japanese regulatory body? Why they are different? What specialty in that? Which has banned? Which is accepted? Which limit you can use it? What is Orange Book? What is inactive ingredients list? What is the maximum daily dose of excipients? See, if you're hearing what is studying so much, what is this? I don't know anything. That will happen, but it's very easy. Once you come to know this, it's very easy. You have to search all these things. You can note down all these words. You have to search it. Get little information in this. What is the different types of dissolution? What are the parameters they will do for a tablet? What parameters they will check for liquid syrups? What parameters they will check for parenterals? All these things you should try to search and get the things. So even in interview also they will ask these questions only, but they won't ask much detail. All what and all I told, no, all these things, minimum knowledge they will ask, minimum basic. What is that? They won't ask you in deep. What is that? What is bioequivalence? What is Orange Book? Those things they will ask. What is pre-formulation? What is stability study? So, as I already told, what they will ask in interview. So you should be confident enough. There is nothing is impossible. Everything is possible. So if you have any questions, please you can ask me. If I know, I will explain you. If anything is need more, then you can please put any questions to my mail ID. It will be displayed at the end of the session, so that I will answer you individually also. Sir, over to you, sir.
Perfect. Thank you so much, Santosh, for providing us an overview of what really happens in formulation and development. I'm sure this is a very, very vast domain to understand in just 1 hour's time. It may not be possible. But what I could understand from this whole presentation is This is a domain exclusively for M-Pharm pharmaceutics. That's number one understanding. Number two understanding is, since this is exclusively reserved for M-Pharm pharmaceutics, no other professionals can try and compete in this profession, right? Unlike other pharmacy jobs wherein everybody is trying to compete for the same profile, right? So here it's only M-Pharm pharmaceutics. There is no other competition. Participants, what do you feel about the information that that Santosh provided in the presentation? Was it useful? Please put in one word in chat box as to let us know, was this information useful? And then also you can ask questions if you have any specific question. We'll definitely try to answer. We are not experts here, but we are still learning. We'll try to answer whatever ways we can. In case we don't know the answer, that's perfectly fine. We'll try to come back to you with proper answers after some time. Is it okay? So let us know in one word, what did you feel about today's presentation? Was the information provided useful? Did you get some idea about F&D, how it works, what are the different departments? And then we will take up the Q&A. I'm waiting for your responses, and then based on that, we'll decide. Yes, participants, you can ask questions in your chat box also. If you have any specific question, please do ask, and then we'll try to answer. You can also raise your virtual hand, and then we'll unmute you. You can answer, ask the question directly to Santosh. Vinit says good and better information. Thank you, Vinit. I hope it was useful and you get some takeaways from this session. Yes. What about others?
There is one question, sir.
All right. I can't see. All right.
How to explore job opportunities for freshers to work in F&D? Because most of the advertisements I found only for experienced candidates. Yes. This even we face it during my time also.
Yeah.
So this is there because nobody will take freshers at the beginning and they will give salary and they will give it, because as a fresher we are having only theoretical knowledge. We're not having practical knowledge. So you have to try it. Even though if it is small company also, you have to join it first, formulation development. Get at least 1 year experience. Then you can join wherever you want, which company you want. But at least to get 1 year experience, at least to join, whether the company is small or big, you first join it. Then you can easily jump into get the knowledge, get the experience. Then you can easily jump into the big companies also.
All right. Any suggestions to fresher to plan their job search in F&D? What is the best time for them to plan the job search? Is it when they're still in the M-Pharm, or is it after completing the M-Pharm? What is your suggestion?
So M-Pharm second year means you are doing projects only.
Yeah.
So suppose if you have completed the project and you are getting the opportunity in formulation development, you can discuss with your college administration, and if the yes, you can join just 6 months early itself, even if company permits also. And because once after finishing the M-Pharm, suppose if you're not getting the job, you have to sit for 2-3 months, and again you will lose some little hope. So if you get the opportunity, you have to utilize completely.
Perfect. Yeah, well said. Actually, in our times, the situation was different. We did not have so much of competition. Right now, the competition has increased definitely. And colleges have also framed a lot of rules, right? During our times, we did not submit the thesis. We get an opportunity. The companies welcomed us. Even the guides were okay with that. In many cases, today, the guides are not okay with that. Guides say, okay, first submit the thesis and then only you try all of that. But my question again is, what kind of preparation, let's say a student is still writing the thesis and from second year, what is the stage and what kind of preparation he or she should do to plan his or her career path in F&D?
See, sir.
Any specific preparation is needed?
Yeah, specific means you're already pharmaceutics department, you're in pharmaceutical department, that itself is specific. And secondary thing is when you're doing second year M-Pharm project, You should do the project which is latest trends. Or it should be very useful to your career. So at least that is the one you can tell during interview also. So that you have the opportunity so that you can tell them, they should say, wow, he has did very good project, he has worked well. So in that also you can easily convince them. And you have to, by studying itself, you should get some knowledge on regulatory and formulation informations. As I told already previously, I guess we should get the things.
All right. Yeah, that's kind of okay. Yeah. All right, participants, do you have any questions you want to raise your hand and ask? Please do that. We do this every Saturday. There'll be one or other learning that is happening every single Saturday. I'm doing this since last 3+ years now. And I really feel enjoyed and overwhelmed to create this kind of awareness. And also learn from people like Santosh, who are industry experts in their own domain, right? I cannot be expert in everything, but it is always good to learn something from these experts who are from industry, right? So if you have any questions, please do ask, because if you ask, you get. If you don't ask, you don't get, right? If you have any questions, you can put in the chat box. Let's learn something by asking.
See, one question is there, sir.
Okay. I don't know. I'm not getting those questions. Where they're asking. They're asking.
Yeah, this is Pallella Naveen Reddy. As per your suggestion, to work in small companies, they are offering less than 20K per month, being studied for 6 years. How far is it good to work for 20K, sir? And some companies are also having bonds for at least 2 years.
Yeah, I agree. Yes.
Yeah, again, it's a competition only, as I told. See, if something advantage is there, again, there are some disadvantages there. Even though it is mainly only for pharmaceuticals person, there is so much competition. See, as you told, it's small company. Small company will be only 20K. As I told, it starts from 2.4 to 5 lakh. It depends on companies only. And at least they're giving this much salary. During our time, it will be still less. Yeah. So after studying 6 years, how far? That's left to you. How competitive enough you are.
Absolutely. So knowledge and competition, we need to prove to the employer, and employer are always ready to, give a better salary to those who are competitive enough. Am I right?
Yes, sir.
All right. Perfect. So if you have any questions, please put to everyone so that everyone can see the questions. I think you are putting the questions directly, so Santosh, I'm not able to see those questions. All right. Anyways, if you have any more questions, please do ask. We are there for another 3-4 minutes, and we'll try to cover up during that time. All right. Thank you, Dinesh, for joining from YouTube. This recording will be available on YouTube, so you can watch later on if you want at your own pace and keep learning from there. If you find these webinars and sessions useful, please do subscribe to the channel and share to many more people, because that's how we learn, right? When we share to others, we learn something from them. And when we get the information from them, we learn something again. All right. So if there are no questions, thank you so much, everyone, for your time. Thank you so much, Santosh, for taking your time, even on a day like this where you could have enjoyed with your family, but you chose to share your knowledge with us. That really means a lot.
Sir, we have to thank so much for you, because you are doing more service. we are giving one speaker itself. So by studying your notes, we have grown. In B form, we have. You can ask any person. Most of the people have referred your notes only. Even though we will do 1 or 2 subjects, but most of the subjects we have referred your notes. So I will advise you, why can't you write books, sir, for the students by referring all the authors' books?
Yeah, actually, many students that told me the same thing, that why don't you write books? I think it will add a lot of value. Yes, something is in progress. I should say, yes, something is in progress. Some planning is happening. I'll let you know very, very soon. But yes, the notes are still in circulation. I happened to visit some college recently, and I found the Xerox copy of some of my microbiology and biotechnology notes that were written in the year 2002. 2000, I think, yeah, 2001 and 2002, right? That was the time I was in second year B Pharm. And those notes are still in circulation. The syllabus has changed, but still the notes are still in circulation. People are you're still using the Xerox copy.
Because so many people, it's very difficult for them to get understand from different authors of books.
Yeah, absolutely. So yeah, that's definitely a need. I think, but I'm not an expert. That's why I never thought of writing any book. Those job is left for somebody who is already expert. But yes, so much of encouragement, so much of suggestions from many students. I'm still thinking, why not do this? Yeah,
Yes.
at least I can read 4 books and write one. That's not a problem for me still. But yeah, definitely I'll plan that. Thank you so much, Santosh, for that. Yes. And participants, stay tuned for more of these webinars. These webinars will happen every Saturday at 6:30 p.m. Next week, we'll have something on medical writing. So if you are somebody who is exploring medical writing as a career plan, then please do join that webinar. And if you want to share this with other friend of yours, then yes, please do pass on that. You can connect with Santosh on LinkedIn. I'll be sharing the LinkedIn profile link of Santosh later on in the groups. Please follow him on LinkedIn and connect with him. If you have any more questions, you may please connect with him as well. And he is more than happy to help anyone get into F&D and support him or her. Okay. So thank you so much. Santosh, you have any final words for this session?
Nothing, sir. I already told everything.
Right, perfect. So thank you so much, Santosh, and thank you so much all the participants, Chaitanya, Grace, Harshida, Ganashri, Kirtana, Krishnagiri, Palrela, Supriya, Vineet, and many of you joining in from YouTube. Thank you so much for your time. All of this encouragement definitely helps me to keep conducting these webinars since almost 3+ years I'm doing this. And, because you participate, I conduct. If you don't participate, then naturally I'll not do it, right? So even a single person gets benefited from this webinar. We are blessed to have you, right? Thank you so much, and keep learning and keep growing. That's the only thing we want to have from you. So stay connected. You'll get the recording on YouTube. Please watch that. And I'll be sharing the LinkedIn profile link of Santosh later on the group. So stay and follow him. You can also search his name on LinkedIn. You can go to my profile and see one of the posts where Santosh is tagged. So follow him and connect with him. If you have any questions, do let me know. So thank you so much. Take care. We'll meet one more time again next Saturday at 6:30 p.m. Thank you.
Thank you. Thank you very much, sir.
Thank you, Santosh.
Thank you all.
Thank you. Take care. Good night.
Good night, sir.